invited lecture session. I hope you all had a nice lunch. I'll be walking around the room at regular intervals, making sure that you are awake. No postprandial sleeping, thank you very much. My name's Lars McNaughton. It's my very great pleasure to invite my colleague and good friend, Professor David Bishop, to come up and present his invited lecture, Training Adaptations, Influence of Nutrition, Genes and Intensity. Before David does that, for those of you that haven't met him, David's Australian. You have to say that exactly right, not Australian, so Australian. He currently works in and lives in Melbourne. He works at Victoria University as part of ICEAL, which is the Institute for Sport, Exercise and Active Living. David's a ex-sports performer. He was telling me the under-8 sack race, but not quite sure how he finished in the under-8 sack race, but he's a competitive individual, so I'm sure he did very well. David's work focuses on understanding skeletal muscle adaptations to training in order to improve sports performance, and that's the sort of thing he'll be talking about today. Would you join me in welcoming Professor David Bishop?
Okay, thanks, Lars. I'm going to get straight into this. So with the Rio Olympics finishing just a few months ago, gives us a really good opportunity to reflect on what went well and what went not so well. And Mark England did a great job at the first day of the conference. And I thought Mickey Yule did a brilliant job last night as well. But first, I just want to get one thing out of the way. So Australia did not have a great Olympics. It was definitely, below our expectations. But as I think, Paul Bryce expertly highlighted, we need to make sure that our data is meaningful. And I think, you all would agree that really the only meaningful way to look at this sort of data is per population. With that, I feel a little bit better. And I'll get on to the presentation. And as I said, I think, after the Olympics, it's a really good opportunity to reflect and see if there's any new ideas out there, new things that we can incorporate into our training. And so with that in mind, what I want to do today is I'm not going to give you this greatest hits, stuff you've all read about and know really, really well and have seen before. What I want to do is something a little bit more like this. Okay, something you haven't heard about before, something new. A lot of the research I'm going to present today is unpublished, stuff that's in our lab that you wouldn't have read before and also what other people have done before. So, the title of my talk you can see there is Training Adaptations, Influence of Nutrition, Genes and Intensity. What I want to do as well, just briefly to pause, is just to thank everyone, Keith, Clyde, everyone for putting on a great conference, bringing me over from Australia to talk. And as I said, I've really enjoyed it. I think it's been a great conference, and I'm really, it's a nice honour, and I'm really appreciative of the invitation to speak here. My focus today is I'm going to use, people might be familiar with this, is Stephen Sealer's polarised idea of training. And it's a little bit different to what I've got here, so I'll show you an example of that shortly. So this is actually, ESSA is the Exercise and Sport Science Australia, so it's the equivalent of BASIS in Australia. And so this was in a textbook published by ESSA. So this is a lactate curve, and this is typically the sort of training zones that are used by Australian sports. So based on, got like the aerobic threshold, anaerobic threshold, there's a bunch of different training levels. So this is, recovery, cool down type stuff. This tends to be sort of continuous, longer, more intensive. Threshold training level 4, interval training between lactate threshold and VO2 max, L5, and then L6 being your sprint interval training. As it's a little bit different for those who are familiar with Stephen Sealer's work, it's a little bit different from his model. So I just want to try and give you an analogy so you can compare it. So if you look at Stephen Sealer's model, what he has is 3 zones: Zone 1, Zone 2 and Zone 3. And by looking at the training programs of elite endurance athletes, he came up, and other people, if you read some of Hageman's stuff in rowing, you see something similar. And so endurance athletes tend to do a predominant amount of training in this Zone 1. So it can be 80-85% of their training. They'll do some training in Zone 3, so this interval type work, and they tend to do very little in this middle zone, so kind of having elevated lactates for a prolonged period of time. So that's going to be my basic model, and I want to go through the research to either back up or dispute some of these ideas. In particular, I'm going to be talking about skeletal muscle adaptation. So a lot of the research you'll see is when we'll take a resting muscle biopsy before and after training, and with various analysis techniques, we look at what happens in the muscle. So as I said, I'll start with some unpublished research, and what we did here is we were looking for changes in citrate synthase with basically any training program out there that we could quantify that looked at changes in citrate synthase activity, and that's typically used as a marker of changes in mitochondrial content. And I think, obviously there's some variability and some scatter, but what was interesting is basically if we look at training intensity and changes in citrate synthase activity, our marker of mitochondrial content, we basically don't see any relationship at all. But if you look over here on the right, considering the data, probably almost was surprising for us, is a pretty robust relationship suggesting that the greater the training volume, the greater your changes in citrate synthase activity, and as I said, our marker of mitochondrial content. And if we look at some of the rodent data, and again we've published this, but we basically collected all of the data that had measured citrate synthase, and a pretty robust relationship. You can see an R squared, so goodness of fit up there of close to 0.9, and again no relationship with intensity, but again suggesting there that the training volume is really key to be increasing this marker of mitochondrial content. And we've also, to test it, we've also done some intervention studies which we've published this year, and basically what we did in this study is we had the participants come in and they did what we've called NT, normal training, for 4 weeks, which was just interval training 3 times a week for 4 weeks. And then we got them doing this intensive phase here for 3 weeks. And what they did in the intensive phase is they did about an hour of interval training in the morning, then they came back and did another hour of interval training in the afternoon, and they did that 7 days a week for 3 weeks. So we've got the megajoules of training, so you can see a massive increase in training volume. And then if we look over here at the changes in citrate synthase activity, this is before the study. After doing their normal training, really not too much change. And then once we did the intensive phase, they had about a 40% increase in citrate synthase activity. And if we go back to this graph that I showed you before, it's interesting. So that's our data point there, which sits nicely on that curve, on that linear fit. So what I would say that, at least in this aspect, I think that there's some evidence out there, and obviously when you train, you're not just only trying to increase citrate synthase activity, there's lots of other adaptations you're looking for, but I think you can definitely make a case that there's something important about high volume training. And so, there's a lot of emphasis at the moment with high intensity training and the benefits of high intensity training, but I would make the argument that there's still a place for high volume training. So I want to switch now to the other side and look a little bit at the high intensity training. And this is just a review that my postdoc and I are hopefully going to submit pretty soon. But basically what we're looking at here is this time changes in mitochondrial function, and what we've got here is going from doing no training to sort of low intensity up towards the high intensity sprint interval training, high intensity interval training. And I think, this isn't sort of mathematical, this is just me using my eye, but there seems to be a trend that, going from continuous training up until high-intensity interval training, we're seeing better changes, this time in mitochondrial function when we do the high-intensity interval training. So I guess, suggesting that the intensity of training is going to be important for getting these specific adaptations, for the adaptations in mitochondrial respiration. And there's other studies that have done direct comparisons here. So this is from a French group, and what they compared was moderate intensity continuous training with high intensity interval training. And what they see there, they matched the two types of training for work, and there was almost 3 times the increase in mitochondrial function, which is this measure here, when they did the high intensity interval training. So in this case, when we're looking at this particular physiological adaptation, the mitochondrial function, it looks like there's an additional benefit from doing the high intensity interval training. We've also looked at this as well, and when we look at mitochondrial function, so this is what we do is we take the muscle biopsy, we put it in a respirometer, and we measure how well the mitochondria can use oxygen to produce ATP, and we see something similar. So when we do the interval training, these are just different measures of mitochondrial function, so don't get caught up with those, but just look at the trend, is that in each case, the black bar, we get quite a large increase in mitochondrial function. This is when we measure the mitochondrial function in the muscle that we're putting inside the respirometer. What we can also do is divide our mitochondrial function measurement by our measure of mitochondrial content, and what that gives us, if you like, is a measure of the mitochondrial function per mitochondria. It's not exactly that, but that's an analogy. And so when we see that, we actually don't see much of a difference. So a lot of this increase in mitochondrial function is also related to the increase in the content. But if we look at sprint interval training, so in this case we've compared changes in mitochondrial specific respiration. So this is how well can each mitochondria use oxygen to produce ATP. We don't see much difference between the moderate and the high intensity. It's only when we do the very high intensity that we do the sprint interval training that we see these improvements in mitochondrial function. And we're still looking at this, but what we're thinking is that with this very high intensity, we think that it's important to turn over the mitochondria. So what we think we might be seeing there is some of the older mitochondria, which maybe aren't functioning as well as they could, are being prompted to be replaced with new mitochondrial proteins, and we're getting improvement in mitochondrial function. So if we go back to this again, so I think we're seeing that there's some, when we're doing the high volume training, we getting some specific adaptations, mitochondrial content, but when we're doing this very high intensity training, then we're also getting the improvement in mitochondrial function. So it's probably not rocket science that you need to do more than one type of training, but I think this provides some support that by having a mixture of both of those in the training program, they may be getting to target different physiological adaptations. I want to try and look a little bit at this zone in the middle. So this is typically doing reasonably prolonged training with relatively high lactate threshold. So it might even be something like in here, for example, doing an hour ride at your lactate threshold. And as I said, it tends to be that most, and the athletic programs that I've seen, that elite endurance athletes tend to not do a lot of training in that zone. And I think what's interesting is when I was working with the Australian team with flatwater kayak, and one day there was a female coach who told me this when we were just having a chat beside the lake, and told me lactic acid destroys mitochondria. And I thought, okay, that's pretty interesting. I've never heard of that before, and never seen anything published about that either. But I think that's one of the good things about talking to coaches, is sometimes they tell you really interesting stuff based on their experience. And not always, sometimes they say some crazy things, but sometimes you look through the literature and there's actually, they can't maybe explain it, but there might be some evidence for that. So we've been trying to do some research and just try and have a look at what is the effects of acidosis on adaptations to training. And so in this case, what we also do in addition to the resting biopsy is we'll take a biopsy when the person has finished their exercise, and we can look at some of the signalling responses in the muscle. And so this was some stuff that we just published this year, and basically what is here, and you'll see this on some of the other figures, I'll just take a little bit of time, is that we take the resting biopsy, then we got them to do their exercise, which was some high intensity intervals, then we took another biopsy, and then 3 hours later we took another biopsy and we measured changes in mRNA. And I think it was mentioned by Ken this morning, so PGC1 alpha is traditionally seen as people call it the master regulator of mitochondrial biogenesis. So if you're doing exercise that you think is going to promote changes in the mitochondria, which should be important for endurance, then you'd expect to see a change in this. So these are rest immediately post plus 3 hours. And we've got two different conditions. So one was a placebo condition, and the other, the grey bar, is that we gave them an acid, just a mild acid, so ammonium chloride, in the evening and also in the morning before they did the exercise. So what the idea was is that they would be doing exactly the same exercise, but in a more, with their muscles having a lower pH or being more acidic during the exercise. And I think it's pretty interesting. So what we saw here is when they did the exercise with the placebo, you see the expected increase in PGC1 alpha, so promoting mitochondrial biogenesis. But when we gave them the acid, we saw most of that increase in PGC1 alpha was blunted. So kind of giving, at least some preliminary support to what the coach was saying, is that if your muscles are too acidic, then maybe they're not going to, the mitochondria, I don't think we're going to be destroying the mitochondria, but maybe the mitochondria aren't going to adapt to the exercise as well. And doing the opposite, I think it's probably a pretty small effect, as you can see here, but this is a study from Marty Gabala's lab, which was published almost the same time, and they did the opposite. So what they did here is they did an exercise training, and this was a resting biopsy in 3 hours post, but this time they gave them either a placebo or sodium bicarbonate before they did the exercise. So this time they wanted to do the opposite and see what happens when you do exercise but the muscles are less acidic. And you can see here they see the opposite result to what we saw, is that with the sodium bicarbonate there was a slightly greater increase, and it was significant in their study, increase in PGC1 alpha. So kind of suggesting if the muscles were less acidic, then they were responding better to the exercise. We've done a few studies on this line as well. So this is changes in training, and what we did in this particular study is we looked at changes in mitochondrial function, and this is a study in rodents, and I have to go at the back and try and get that video to play. But what we did here, because it's a bit hard to give the mice a glass of water and with the sodium bicarbonate and ask them to drink it, so with a gavage we actually put the sodium bicarbonate directly into their stomach before they did the task, and then we put them on the treadmill. And I think one of the interesting things with this experiment is then, so what we've got is the two rats running side by side on the treadmill, doing exactly, obviously the treadmill, it's one treadmill, so the treadmill speed's going the same for all of them. One of the rats had the placebo, so just some water, before they did the interval training. We did interval training with them. And then one of the other rats had sodium bicarbonate before doing the same identical training. And just concentrate on this. So this is the mitochondrial function in the soleus muscle using pyruvate as a substrate for the mitochondria. And this is just the control rats who did nothing. As you'd expect, the placebo group after the training had around about a 25% improvement in their mitochondrial function, and the other group who did exactly the same training, but the only difference is they had sodium bicarbonate before every single training session, had around about double the improvement in mitochondrial function with training. So again, matching up with what we'd shown earlier, is that maybe doing too much training and making your muscles too acidic you may not get the optimal skeletal muscle adaptations out of that approach. And I'm sure, rats are interesting, but I think you guys are probably a little bit more interested in humans. We haven't done exactly the same study in humans, but we did a study without measuring mitochondrial function in humans. And basically, so what we did here is they did interval training. These were females, sports science students. They did interval training for 8 weeks. And again, we had them side by side doing exactly the same training. And the only difference was that one group took sodium bicarbonate again before every single training session. And what you can see here is their time to fatigue at VO2 max, which was our endurance test in this particular study. Started off pre-training, we matched them and we randomised them, so they started off exactly the same. Both groups improved their time to fatigue or endurance, but the group that took sodium bicarbonate before every single training session had a significantly greater improvement in their endurance test, their time to fatigue at VO2 max. So again, I think a bit of a consistent picture emerging is that potentially if we can minimise the acidosis during training, then potentially we can get greater adaptations to training. When we looked at some of the physiological responses, we didn't see any difference in VO2 max, and maybe that's because the VO2 max also reflects the central adaptations. When we looked at the lactate threshold, which I think is a better marker of peripheral skeletal muscle adaptations, we saw again that the group that took sodium bicarbonate had a significantly greater improvement in their lactate threshold. We've also seen some other markers, so it's not always consistent, but in this case we looked at some of the lactate hydrogen ion transporters in the muscle, and this was the previous study I showed you before in the rats. But if we just look in the MCT4 in the middle, again, the placebo group had an improvement in their lactate transport out of the muscle, or their transporter responsible for the movement of lactate out of the muscle, and the bicarbonate group had a significantly greater improvement. So better adaptations in this particular protein important for pH regulation when we minimised the change in pH during training. And we've also recently done this with humans, and so this is some unpublished stuff, but again, this was just a response to a single bout of exercise, and this was a typical cycle training session where they did 30 seconds all out, had 20 minutes rest, and repeated that a number of times. The placebo is in white, and what we saw is that after this high intensity training session, there tended to be a decrease in the MCT1, so the proton transporter, but the group that took sodium bicarbonate had an increase in this transporter immediately post-training, and this was 24 hours later, and this was significantly greater than what occurred in the placebo group. And this is actually going back a little bit, so this isn't one of our studies, but I think this is interesting as well. This is Nigel Steptoe did he's with us, but also did his PhD with John Hawley. And what he did was he compared, and these were in pretty good cyclists, compared a bunch of different types of training programs. It was only for 3 weeks, and you can see the characteristics there. So number of bouts, how the duration of the work bout, the intensity of the work bout, and the rest between the work bouts. And this was the percentage change in VO2 max with the different training programs. What was interesting for me is I then went through and calculated, and this is in no particular units, but so this red line represents the total volume of training done in both of those groups. So even though the characteristics are different, the volume of training tended to be different. And you can see here that basically the changes in VO2 max tended to follow the volume of training that the participants did, except for this last one on the end. So here they're doing 8-minute bouts at around about what you'd expect to be the lactate threshold with very little rest in between. So in this case, you'd expect, and it wasn't reported in the paper, but they would have been training with a relatively high lactate value for the entire training duration. And so the volume of training was similar to here, but the adaptation to training is much less. So maybe I'm sort of wanting this to be consistent with our hypothesis. But I think that kind of supports, again, the idea that maybe doing too much training where there's accumulation of lactic acid may not be the best approach. So if we go back to, again, the sealer model. So I think, not saying don't do any training at threshold, but I think, an argument can be made that maybe the best adaptations won't be achieved by doing too much training that involves high levels of, prolonged exercise with relatively high levels of lactate. And I think the other thing, two considerations with this as well, and I'm not necessarily advocating sodium bicarbonate during training, but I have, speaking to some coaches and athletes, I have heard that some athletes do try this approach. So, potentially that could be, something for some athletes to consider in sort of a periodised training program, is during some of the more high intensity training where there is likely to be a greater decrease in muscle pH, to maybe consider incorporating sodium bicarbonate. And I think getting away from, the supplement idea, I think this is maybe also one of the importance of the low volume training as well, and getting a good base, because I think what can happen is athletes who can do a fair bit of training and get this base, they will move their lactate threshold to the right, which means that they'll be able to do relatively high intensity training with lower levels of lactate. So that may be also one of the other benefits of the importance of this low volume training, is it also primes you to get better adaptations when you do the high intensity training. I'm not going to do a lot of talk of this, but I just want to talk briefly about some of the role of manipulating nutrition around training as well. And I saw there's some good posters on that here as well, and particularly this idea of doing training with low carbohydrate. And so there's quite a few studies. Henriette Pilgaard in Copenhagen did some of the early works, and it came up with this this train low model. So the idea that potentially training with low muscle glycogen levels, you may get better adaptations to training. And I think that the evidence is probably a little bit mixed, but this is one recent study which came out of Kent Sarlin's lab, and what they did here is they got them to do a glycogen depleting exercise the day before, and then they either gave them carbohydrates or didn't give them carbohydrates. So they would then come back in the white bar in a normal glycogen condition or in a low glycogen condition, and then they did their continuous exercise, a single exercise training session, and then they took another biopsy afterwards, and they looked at changes in, again, this PGC1 alpha, so this master regulator of mitochondrial biogenesis. And what you can see here is that they saw that when these are reasonable athletes as well, when they did their training with low muscle glycogen levels, that they saw that a greater increase in PGC1 alpha mRNA. So suggesting that doing their continuous training with low muscle glycogen levels may amplify the response to training. And I think there's a couple of just want to make a couple of observations here. One is that the change in PGC1 alpha in the normal glycogen condition was quite low. I'd probably expect a little bit higher, but anyway, so there's probably in this type of exercise mode, maybe there's more scope for a nutritional intervention to amplify the signal. And again, I'll just show you some unpublished data from our lab, and I'll explain this, but basically we didn't see the same thing when we did high intensity exercise. And so I'll go through explain these results, but we're thinking that maybe with the high intensity exercise, because you may be already getting close to a maximal PGC1 alpha response, then maybe it's hard to go even higher than that maximal response. For this particular study, this was a PhD student from Brazil. And it's a really nice design, I think. If you read through the low glycogen literature, one of the problems is that a lot of the studies that claim to be low glycogen are also training twice a day. So what they do is they do an exercise in the glycogen depleting exercise in the morning, then they'll either give you carbohydrates or not give you carbohydrates, and then they'll do another exercise task in the afternoon. And so if you see an effect, it's a bit hard to know, is it because they've got low glycogen, or is it because they're just training twice in one day? So we wanted to try and separate those two effects. So what we did is that we had them do a high intensity interval training session, and so this is the biopsy before, this is the biopsy immediately after, and this is the biopsy 3 hours post. And there was 3 conditions. So the grey is that they just came in, and in the afternoon they did a high intensity interval training session. The white is a once daily, and so what they did in this condition is the evening before they came in and did a glycogen depleting exercise, then we didn't give them any carbohydrates overnight or in the morning, and then in the afternoon they came in and did the high intensity interval training at the same time of day as the control group. And the third condition was the twice a day, so the black. So what they did in this condition is that they came in the morning, they did their glycogen depleting exercise, then we waited a couple of hours, and then they came back in the afternoon and they did the same high intensity exercise at the same time of day. So hopefully what we're trying to do is to try and distinguish, is the effect due to having low muscle glycogen, or is the effect due to doing 2 exercise sessions in one day? And our results would suggest, at least with the high intensity exercise, it's not due to the low glycogen. So if you compare just the white and grey, the only difference is the muscle glycogen levels, and you can see there we don't see any significant difference between those two conditions. What we do see is what other people have seen is that when we do the twice a day training, what we see is a greater response of PGC1 alpha, so suggesting a better activation of mitochondrial biogenesis. And so what we think might be happening is that what you're seeing, and sorry, I'll do some hand actions here, but so what we think you're seeing is when you do the exercise in the morning, you're getting an increase in the PGC1 alpha response, and then you're starting off at this higher level, which you can see here. So this is immediately before the afternoon exercise session, and then when you do the next session, maybe you're getting superimposed on that already elevated PGC1 alpha levels. So we think that, if you look through the literature, especially with high intensity exercise, it's probably the fact of doing 2 exercise sessions in one day rather than the low muscle glycogen, which is important. So if we come back to this schema, and I think it was mentioned this morning about periodizing some of these things. So maybe with the low intensity, the zone 1 type training, maybe it's worth considering doing some of those sessions in a low glycogen state, but probably going to be less helpful if you're training in zone 3. But when you go about periodizing your training program, maybe consider sometimes trying to do that. And this is where, I'm sure some of this stuff people are going to say, oh, yeah, coaches, athletes are already doing this already, but I think it also makes sense is to sometimes to pair continuous exercise fairly soon after with a high intensity training session, so that you might be able to amplify some of these mitochondrial responses to exercise. And I think what's going to be reasonably important, because some of these markers respond really quickly and then turn off, so PGC1 alpha will increase, probably reach a peak maybe between sort of 2 and 5 hours post-exercise. So it's probably a consideration on how you program your training, is that if you do the continuous exercise very early in the morning and then the interval training late in the afternoon, you're probably going to lose that effect. So it's going to be important to maybe try keeping those reasonably close so that you can amplify the response to training. Really great session this morning talking about the recovery strategy, so I'm not going to spend too much time on that, but obviously the other way, in addition to, is nutrition, and also there's aspects to do with nutritional recovery, but also the use of cold water immersion as a post-exercise, using it post-exercise to try and promote adaptations to training. And so this is a study from Isan and supervisor Chris Abyss, who's in Perth in Western Australia. And so what they did here is that they had two conditions. So they did an exercise session. This is again resting, post-exercise, and then 3 hours post. And they did exactly the same exercise, except one condition was just a control condition, and the other one they did cold water immersion. And what they showed in here is that, you can see here there's a pretty big variability, but when the cold water immersion was after the training session, they got a greater response of PGC1 alpha. And again, you can see here that the response of PGC1 alpha is pretty low to this type of exercise. And so maybe with this, more moderate intensity exercise is a greater scope to amplify the response. And if you look through the literature, I think, there's some good scientific evidence to support this potential, and there's some interesting research actually in fish as well. So fish, when they're in really cold climates, that will promote their mitochondrial biogenesis as well. So I think, there's some other support for that. And we've, just in review at the moment, this particular study, we're looking at more higher intensity. So this was using sprint interval training, and then either doing the control condition, or black cold water immersion as before. And we didn't see too much difference. There's a trend with the PGC1 alpha for the cold water immersion to amplify the response, but this time it wasn't significant, unlike the ISAN study. And I think, as I mentioned previously, I think maybe with the high intensity exercise there may be a little bit less scope for the cold water immersion to amplify the response. But I won't go through this in detail, but we also looked at some other markers of mitochondrial biogenesis, some other proteins changing in the muscle. It wasn't always significant, but I guess there's a bit of a trend for the cold water immersion to be better, and I guess in no case was it worse, the cold water immersion. And if we look at a more practical training, or practical model, so this isn't changes in skeletal muscle, this is from Shona Halson in the AIS. In this case, she was looking at a 4-minute cycle test, and they did their 3 weeks of training, either, again, white control or black with cold water immersion after each training session. And this was in pretty good cyclist training at the Australian Institute of Sport. And they actually saw a greater improvement. It wasn't, it's not massively greater, but a slightly greater improvement in this 4-minute cycle time trial test in the group that had cold water immersion after every training session. They also measured some other performance changes, and most of those changes weren't significant but probably trended towards the cold water immersion group. So I think, again, just returning back to this schematic, so I think, I can't really, at least with, as was mentioned this morning, at least with the continuous, or at least with endurance type training, I haven't found anything to suggest that you're going to get a negative adaptation by having cold water immersion after a training session. So it's probably something to consider overlaid on top of this training model, is adding some cold water immersion after the training session. And as I said, if at least the evidence to date suggests that it's unlikely to be detrimental. I wanted to also, so this was, this is based on some of Ed Coyle's data, but I just wanted to highlight a few things. One is why I think this type of training approach is important. And so this, as I said, this is some data from Ed Coyle, and what they've got here is they had a group of pretty good cyclists, well-trained cyclists, who had the same VO2 max but a different lactate threshold. So you can see here the blue is the high lactate threshold, and the red and purple is the low lactate threshold. And so I just want to highlight here that if we were training everyone at the same absolute intensity, or even at the same relative to VO2 max or heart rate intensity, these guys here are going to be training, so this is the scheme that I showed you that we use in Australia, but these guys are going to be training at level 2, and so that's the low volume type training, whereas these guys are going to be high up L5, so the interval training intensity. So you probably, you wouldn't expect that both of these are going to get the best adaptations, even though they're training at the same intensity. So that's why I think that type of schema and using the lactate threshold is important for regulating training. The other thing that's, and I think most people know this, but I think it's nice sometimes to see the science to support this, but is that the muscle adapts very quickly to any stimulus. And I think what this shows is the importance of, as the lactate threshold and as the fitness of athletes improves, is to alter that training stimulus. So what we've got here is the resting biopsy, and again looking at PGC1 alpha, response to the first training session, and then it comes back after 24 hours. Then we've got the third training session here, then the fifth training session, and then the seventh training session. And so what you can see is each time that the participants in this study were doing exactly the same training stimulus, but each time they're getting a less of a response to the same training stimulus. And we've also seen something, some similar with our unpublished stuff. In this case, we're looking at PGC1 alpha in the nucleus, but what we've got here is the response to the first time they come into our lab. Big response, they do the exercise, you get exactly the sort of molecular response that you'd expect, PGC1 alpha moving into the nucleus and initiating transcription. After 3 weeks of training, they came back, and we've got the black bars there, and we see basically nothing happening to exactly the same exercise. So I think just again highlighting the importance of altering the relative training intensity as people get fitter. And just concentrate on it. You can pick basically any column. Let's just pick this one. But this is the one I showed you before, but with an extra data point. So this is pre-training, the normal training. Then we did the high volume training for 3 weeks, and we got around about a 40% improvement in their mitochondrial function. What we then did is gave them 2 weeks where they didn't do any training, or they basically didn't do any training. And in each case, the mitochondrial function basically returned exactly where it was before. So I think this is, a couple of things. One is it highlights the plasticity of the skeletal muscle. So when you do training, especially in untrained or moderately trained people, the muscle responds really quickly, but as soon as you remove that stimulus, it goes back to where it was before. And so I think this is also then becomes one of the challenges is when you're doing it, trying to prescribe a training taper, is how do you get the best out of the taper so that you're removing the fatigue component and hopefully maximising performance, but it really doesn't matter what you do, or it does, but probably you're also, during a taper, you're going to lose some of those physiological gains. So how can you best construct the taper, and I don't have the answer today, but how can you best construct the taper so as to remove the fatigue, but also being careful not to remove those hard-earned physiological adaptations. I'm just going to finish on one last aspect, and I gave a talk at Loughborough on Monday, but I guess just the genetic component is becoming increasingly studied, and as I said at Loughborough, I think we're a fair way from being able to use genes to modify training and to prescribe training, even though there's some companies that are trying to do that at the moment. So I'm just going to highlight a little bit of our research where we've tried to look at the influence of certain genes on the response to training, and this what's quite often done in at least in the lab is the use of a knockout model. So if you haven't come across this, so basically what it is, if you want, if researchers want to know if a particular gene or a particular protein is important for the response to training, what they can do is they can inject some genetically engineered cells into the mouse, and then they breed a number of generations, and then ultimately you end up with a mouse that lacks a particular protein or gene that you want to look at, and then you could get them to do training or whatever and see if that affects the response to training. I'm not going to talk about that, but what would be really cool and really unethical is to do the same thing in humans. And to a certain respect, we've done that. And it's not, we've got ethics approval and everything. But what I'll go through is what we've tried to do is we've taken advantage of some natural human knockouts that exist in the population. And I'll show you specifically, but what we've looked at is what's known as a speed gene. So so it's known as ACTN3. And so if you look around, the room here, about 20% of you won't have this speed gene, this particular protein. And there's some evidence that this may affect the response to training. So we've done some research doing this, and I'll go you through, it's pretty complicated, or not complicated, but time consuming. So we need to get a lot of people to express an interest. A lot of those won't be eligible because we've got really strict entry criteria, and then those who are eligible, not all of them will agree. We then separate them into the 3 genotypes, and what we want, so we get rid of all these RX guys, and we just want the RR, who are the ones who have the protein, and the XX, the ones who don't have the protein, and then we get them to do into our lab and do some exercise. The first thing they do is they come in, have a resting muscle biopsy, they'll do the exercise, and we're doing some interval training. And then at the end of the interval training, we'll then take another muscle biopsy, and then, as per usual, we'll wait 3 hours, and then we'll take a third muscle biopsy. Then we'll put these guys into a training program, so they're training 3 times a week for 4 weeks, and then we take another final biopsy. And so I'm just going to show you some of it's still only preliminary data, so we've only got around about 15 in each group, and we're aiming to have about 40 in each group so it's going to be probably a 3 or 4 year study. But I think some of the results are interesting. So what we're looking at here is, in each of the next figures, is the RR. So they're people who don't have the protein, who do have the protein, and XX is the people who don't have the protein. So it's been naturally knocked out. And as I said, that's naturally happening in the people around the room. And so again, looking at PGC1 alpha, and we've just done some more participants, and it's actually significant now, but what we see is that people who don't have this ACTN3 protein have a better response to PGC1 alpha, to a single interval training session. And if we look at the changes with training, it's not significant, but there's a nice trend there that matching the PGC1 alpha is those who lack this particular protein have a greater increase in citrate synthase with training. And if we look at mitochondrial function, quite a lot of variability, but this is significant this time, is that the people who lack this protein are also getting a greater improvement, or they're getting a significantly greater improvement in their mitochondrial function in response to doing exactly the same training session. So this is just example of one gene that we're looking at which we think is going to influence adaptations to training. It's probably going to be a small influence, but because we can genotype these, when we get towards the end of the study, we'll be able to do, probably not the full genome, but we'll be able to look at thousands of genes and see if there's other genes that in this cohort, like I said, aiming for around about 100, 150 people, if there are other genes that are predicting the skeletal muscle response to training. So again, just returning back to this, so all those factors, and then ultimately, I think there's also, with the future research, we're going to be able to see the influence of genes on the response to training, and probably also on how we need to periodise training for people with a different genetic background. I'll just return to Sealer's stuff. So he tweeted this a couple of weeks ago, but I think I like this model, and I think it matches up, at least from some of the stuff that we're doing, with what's occurring in the literature. So I think this is your typical pyramid. This is the stuff that I think there's pretty good evidence for, and also probably the key stuff. getting the right volume of training, the right intensity of training, and also this distribution. So having the training program which has the right amount of high volume and the right amount of high intensity training. And at our ESSA conference, as I said, the equivalent to BASES in Australia, we had the conference in April this year. Stuart Phillips was there, and he talked about training being king and nutrition being queen. So I think, the training is rightly so at the bottom of this pyramid. And then there's probably some of these other things that we can then, kind of like the icing, I think the words of this morning were icing on the cake, that we can maybe do to try and optimise what's happening here. And I think, spending all your time getting this right is probably not going to get you the right outcome unless you got this as good as possible. So he said some of these things that I mentioned, so enhancing the training stimuli. So I think there's some evidence there. So incorporating some altitude training, maybe some heat training, maybe sometimes using low glycogen. Also looking at cold water immersion to promote the recovery from training. So the tapering and the pacing strategies. But I think this is the key, and I think the getting this right is going to be more important than the other aspects. So I just want to, I'll finish up there. So I just wanted to, this is my research group. So obviously anyone who works in the lab, obviously most of that data was generated by my PhD students and postdocs. So I want to thank them for all their hard work. And this was actually we had a great Gatsby themed gala dinner at the ESSA conference early this year, and so that's my family up there. So just want to thank them for all their support and letting me come to conferences like this when I should be home mowing the lawn or whatever I should be doing. And so thank you very much for your attention today.
We have about 5 minutes or so just over for some questions to David. So thanks for a great talk, David. But do you have any questions around the room? One in the front here, David.
Yeah. Hi, Simon Wegger from iFleet. Something I've pondered for a long time, although I'm not a physiologist, is that if adaptations, such as mitochondrial ones, are very hard won, but easily lost, do you think that has an evolutionary purpose? Is it, for instance, just to slightly improve resting energy expenditure, or do you think that there is a grand design behind it?
Yeah, no, it's a great question, a bit philosophical for this time of the day. But no, I think it's just one of those things, as I mentioned, that the muscle is really malleable or plastic. So when there's a change in the demands that are placed on the muscle, it will quickly respond. And then the same, when those demands are removed, it will quickly, that stimulus will go away. But I think I didn't talk about it, and we haven't done too much research on it, but I think one of the interesting things also is then this idea of muscle memory. So I think there's some evidence that maybe there's some epigenetic changes, so that when I present it that way, it kind of looks a little bit gloom and doom. Okay, I'll train, and then as soon as I've injured for a week, I've lost everything. But there's some evidence that because of these and potentially epigenetic changes, is that when you go back and do, if you're sick for 2 weeks or whatever, then you go back and do another training session, that you'll respond more quickly than what you did the first time around. So I think, I don't want to be too gloomy about, every time you train, it's going to be, as soon as you have a break, you're going to be wasting all that. But I think, definitely the muscles, everything that we see, and that was just the mitochondrial example, but the skeletal muscle is really plastic, and as soon as you put new demands on it, it will respond. As soon as you take those demands away, then it will go back to what it was before, because it doesn't need that extra capacity anymore.
David, how are the elite athletes, the Wigginses of the world, using this sort of data and their coaching teams? Are they using it to advantage? AIS, for example?
Yeah, I think sometimes I'll present these to coaches, and I really enjoy that because I think I show that example of the coach saying lactic acid destroying the mitochondria. So I think it's good to get that feedback. And at the ESSA conference, I did a session where I presented some of this data, but with Philo Saunders, who's a physiologist, but also he coaches some Olympic athletes. And it was interesting. So he put up a training program, and to be fair, it was reasonably similar to I think what the science would recommend. And I think some things that I wouldn't be doing research if I didn't think we could improve some of those training things. But I think by and large, I think a lot of the training programs, you've got the mix of the art and science, but I think they're based on the literature, but also years of trial and error that probably ends up being pretty similar to what you would recommend as a sports scientist.
Hi there, Dave. Thanks for a great presentation there. A question about the genetic influences that you picked up on at the end there. I guess given the number of potential candidate genes that are going to have an influence on the adaptation to training, and of course the crosstalk and the potential interference between them, how far away do you think we are from being able to use genotypic data to actually be able to influence the training program more than perhaps coach, athlete, intuition, self-understanding and so on?
Yeah, it's a good question. I'd be really surprised if it ever becomes more than the coach intuition. I think all these things are, tools that, if you do some sort of physiological test in the lab, I think, good coaches, you end up kind of confirming what they tend to believe anyway. But I think there's still going to be a place for that. And there was, I think, Claude Bouchard is the guy who's, the father of this area. So it was back in, I think, 85 or something like that, where he did his big paper. And I think he would have, first looking at some of these influences, and I think back then he would have been thinking he'd be sitting back retired and enjoying the fruits of his research, and we'd have all these amazing genetic markers. So I think we're definitely not there yet. There was an interesting study published in kind of a Polish journal, the Biology of Sport, where they looked at, and got a little bit of criticism, maybe a little bit unfairly because it was also linked to a genetic company, but basically what they did is that they assigned people to two different training programs, either based on their genotype or not on their genotype, and the result was strength training, and the results were that doing training based on your genotype, they got a better response than doing training that wasn't based on their genotype. So I think that's the first example that I've seen of that. And I think, like I said before, I think getting the training intensity and volume and all that sort of stuff that's going to be the base of the pyramid, and then maybe using the genes to tweak the training response a little bit is going to be a little bit of that icing on the cake. But I think if you don't get the training volume, intensity, and distribution right, then all the gene information in the world won't help. And I think just a last point on that, I think whenever you talk to anyone working in genes, they're always pretty hopeful that soon it's going to happen. It seems to keep saying the same distance away. But I think, when they first sequenced the genome, I think it was like a couple of $1 billion to sequence the whole genome. And, now it's $1,000, and they're thinking it's going to be $100 in the next few years with improvements in technology. So I think that's been one of the limitations, that people have tended to look at candidate genes. And I think, it's pretty unrealistic that there's going to be one gene, and it'd be a pretty, from the question before talking about evolution, it'd be a pretty crappy evolutionary design to put all your eggs on one gene. So, it's always going to be multiple genes, and I think, we know that genes influence performance, they influence the response to training, and I think maybe with the improvements in technology, we'll start to get a little bit more insight into what might be some of the more important genes.
One last question at the back, and I think David will be available after for coffee if you want to have a chat. One last one at the back.
Can I just ask your change from the moderate to heavy intensity zone on the light diagram? Was we using 0.4 millimoles above baseline, or was it Edward Coyle 1 millimole from the deer?
Yeah, so we're doing some. It's one of the big problems, because I keep, you talk to people and they'll say, okay, yeah, this study did, or this athlete's training above or below the lactate threshold, and when you go through, there's probably about 100 different lactate thresholds depending on the protocol and the actual method. So we're doing some research to try and standardise that. But to your question, so it's based on the AIS. So basically, yeah, it's not based on the coil model. Is they use a 0.4 increase as the aerobic threshold, for want of a better word, and then they'll use the modified Dmax for the lactate threshold.
The ADAPT program.
Yeah, using the ADAPT. Yeah. So it's a bit didn't line up 100% perfectly with the sealer model. So some people use they use it sort of more based on the ventilatory threshold, but reasonable approximation that you've got that low intensity moderate where there's not too much change in lactate, and then you've got the other where the high intensity interval training, sprint interval training, where you're starting to get reasonably high lactate levels.
Ladies and gentlemen, we are out of time. Would you join me in thanking David for a very nice talk? Thank you.